Photosynthesis is a solar energy-fueled conversion of carbon dioxide and water into carbohydrate and molecular oxygen. Early research into photosynthesis focused on the evolution of oxygen and its source. Initial hypotheses claimed the breakdown of carbon dioxide to be the source of evolved oxygen. However, the mechanism was not understood. In the mid to late 1930s, Robin Hill demonstrated in a set of experiments that oxygen was produced by chloroplasts under illumination and in the presence of water. Hill added small amounts of muscle hemoglobin to a chloroplast extract solution for its ability to shift its maximum absorbance wavelength by combining with available oxygen to form oxyhemoglobin. This property of muscle hemoglobin allowed Hill to spectroscopically quantify oxygen evolution in vitro. Today, adaptation of Hill's experiment allows for quantification of electron transport along the electron transport chain by utilizing several artificial electron acceptors other than muscle hemoglobin. In the method presented here, dichlorophenyl indophenyl sodium salt, or DCIP, will be utilized to indicate electron transport in the presence and absence of a photosynthetic inhibiting herbicide. Dichlorophenyl dimethylurea, or DCMU. Other reagents included will be distilled water and a biological buffer. DCIP is a useful indicator of electron transport. Upon reduction by electron transport, DCIP will change its color from its oxidized form of blue to its reduced form, colorless, allowing for spectroscopic measurement of electron transport. To begin the experiment, a tris NaCl buffer of specific concentration is prepared by adding 1.02 grams of sodium chloride and 0.12 grams of tris buffer to 50 milliliters of water, forming a 0.35 molar sodium chloride and 0.02 molar tris buffer solution. The buffer solution is then titrated with concentrated hydrochloric acid to a pH of approximately 7.5. Once the desired pH is reached, the buffer solution is transferred to an ice bath. To begin the chlorophyll extraction process, a pre-chilled mortar and pestle is used to homogenize 4 grams of spinach leaves, of which the majority of major veins have been removed, 100 grams of purified sand to act as an abrasive, and 15 milliliters of buffer solution, which was prepared earlier. The resulting suspension is then filtered through 4 layers of cheesecloth into a small beaker. The pestle is used to press any remaining suspension absorbed by the cheesecloth into the beaker. The filtrate is then transferred to a 15 milliliter centrifuge tube for further chloroplast isolation. Filtrate is then centrifuged at 200 G for 1 minute to remove any remaining plant debris. Resulting supernatant is decanted into a clean chilled centrifuge tube. The chloroplast fraction is then isolated by centrifuging at 1300 G for 5 minutes. Remaining supernatant is discarded and the chloroplast fraction is returned to the ice bath. The isolated chloroplast fraction is then resuspended in 10 milliliters of the tris NaCl buffer. To maximize solution uniformity, the centrifuge tube is sealed and inverted several times, then returned to the ice bath. The chloroplast solution is then diluted by combining 4 milliliters of chloroplast solution with 6 milliliters of tris NaCl buffer in a test tube wrapped with aluminum foil to prevent the incident exposure of the extracted chloroplast to light. After adequate mixing, the diluted solution was returned to the ice bath. Next, a 10 to the negative 4 molar solution of DCMU was prepared by combining 3 milligrams of DCMU with 50 milliliters of water. A 4 times 10 to the negative 4 molar DCIP solution was prepared by combining 6 milligrams of DCIP with 50 milliliters of water. These solutions are used as an inhibitor of photosynthetic electron transport and as an indicator of photosynthetic electron transport, respectively. A solution containing 1.75 milliliters of the tris NaCl buffer, 250 microliters of the DCIP solution, 250 microliters of distilled water, and 250 microliters of the chloroplast suspension was combined in a cuvette wrapped with aluminum foil to prevent the reduction of DCIP by illuminated chloroplasts. Serving as a control, the cuvette was placed in a 20°C water bath for 10 minutes, at which point absorbance of the solution at 600 nanometers was measured, and absorbance value recorded. A similar solution to the control was prepared with the exception of wrapping the cuvette with aluminum foil. Absorbance of the prepared solution was measured immediately, then placed in the water bath under photosynthetic light. The cuvette was removed and absorbance measured at 1-minute intervals for 10 minutes, and absorbance value recorded. The spectrometer was re-zeroed after every 5 readings. Finally, a solution containing the photosynthetic inhibitor, DCMU, was prepared and absorbance values measured at 1-minute intervals as described earlier. Absorbance values were recorded and compared to absorbance values of the